Okay, so this is discussing a concept known as endosymbiosis. Obviously, you have the prefix endo, which everyone by now should know means in, right? And symbiosis just means close relationship. Not good, not bad, just a close relationship. So those are the two parts of the word that are important for understanding the meaning of endosymbiosis. So about 540 to 600 million years ago, MYA, 540 to 600 million years ago, we have evidence of the first eukaryotic cell. Now remember when we have evidence of the first life on Earth was about 3.5 billion years ago. So 540 to 600 million years ago is the first eukaryote. But 3.5 billion years ago, right, first prokaryote. So obviously prokaryotes came before eukaryotes and by a long time. And we know that there were we know from the fossil evidence, and all this is from the fossil evidence. We can see this in the previous pages when we discussed the fossil evidence of cyanobacteria. Remember, cyanobacteria are prokaryotes that can do photosynthesis, right? So photosynthesis they're prokaryotes. And they form those piles of stones that we looked at in the fossil evidence on previous pages. So when you're talking about cyanobacteria, you're talking about the precursors to what we call today mitochondria and the chloroplast, the two organelles that we studied earlier when we looked at the cell. So when you look at chloroplasts, we can see that there's two membranes. And let's look at chloroplasts first, and then we'll look at and the reason I want to look at chloroplasts first is because that's what you see here in the chloroplasts in the discussion. The first thing you look at here is a chloroplast. Next to it is the mitochondria. There are other organelles in different structures that may have formed in this way, but these are the two important ones, the chloroplast and the mitochondria. So let's look at the chloroplasts. And when we look at chloroplasts, the interesting evidence that supports what we're about to talk about is there's some evidence that kind of leads to a conclusion, an explanation. So we have some evidence we have to find an explanation for. Let's look at that evidence. First, a chloroplast has circular DNA. You know what? Why don't we just do it this way? Why don't we say chloroplast? Why don't we do one of those Venn diagrams and compare chloroplasts and mitochondria and see how we might be able to. So chloroplast, mitochondria. draw that. Let me make this smaller so that we have more room. I know they're awful circles, but you know I'm not the greatest artist, so it'll have to do. All right, so when we talk about chloroplasts, well, one thing they know is they do photosynthesis. They're found in plants. Uh, they convert light and CO2, carbon dioxide, and water gets converted to sugar. Right? Mitochondria, on the other hand, take sugar. They take sugar. Well, let's write down what they do. They do respiration. They do cellular respiration specifically. So respiration, I'm sure you know, is just breathing. Cellular respiration is what the cells do, and they take sugar and oxygen, gas, which is why you breathe when you respire, and you take and make, and the mitochondria make CO2 and water. So you see the reverse reactions. And they're found in all eukaryotes. I hate using the word all. I'm sure somebody's going to find some exception where you're not going to find any mitochondria, but honestly, I can't think of one. All eukaryotes. Have mitochondria. So, all right. So that's a really big, that's what we see. This is evidence, right? These are facts. So then the next thing is, what do chloroplasts and mitochondria share with each other? Well, or don't share, what do they share, I guess is what we're going to start with. Both mitochondria and chloroplasts have double membranes. That are like those found in prokaryotes. The sugars and the components and the lipids found in the membrane, internal membranes of mitochondria and chloroplasts, are like those found in prokaryotes. They have circular DNA, like that found in, like those in prokaryotes. Then the other interesting thing about both of these is that they have now. When I say they have circular DNA, you have to understand these are organelles. The mitochondria specifically are organelles that are in every cell of your own body, and your DNA is the DNA that makes you who you are, that makes you different from all the rest of the creatures on the planet. It's inside your nucleus. Well, this mitochondria, this organelle, which is many of them, as you know, inside each cell, has its own DNA. To do its own, they replicate on their own. They do binary. They do their own they do their own replication, organelle replication on their own. They do their own DNA synthesis. They do all that on their own. If, for lack of a better terms, I'm not sure what term you'd use, but they do binary fission within the cell, getting ready to do cellular division. As we looked at the cell cycle, they multiply themselves in your cells right during during the first stages of the cell cycle before M phase. So that's really weird and really interesting that happened. No one expected that to be the case, but that's the case. And then when we look at both of these organelles, and of course chloroplasts do that in plants, right, where mitochondria are found in plants and animals and all eukaryotes, right? Okay. And then they also have their own ribosomes. That are like prokaryotes. All right. So they have double membranes, and the inner membrane is like that of prokaryotes. They have circular DNA like those of prokaryotes. They have their own ribosomes, and the ribosomes are more like prokaryotes than they are eukaryotes. And yet they're found in eukaryotes, mitochondria and chloroplasts. Now let's think about when they formed, when they would have formed, because this happened again 540 to 600 million years ago. Well, what happened before we found them? When we look at the fossil record, remember what we're talking about. We're looking at these layers of Earth, layer after layer, laid down by time and weathering and etc. events. We look here, these are eukaryotic organisms. We find eukaryotes and prokaryotes. You know what? Just because it'll make it easier. And we're talking about, by the way, I want to make sure you understand, we're talking about layers and layers and layers and layers and layers. Layers and deep, deep, deep into the Earth's history. If we say P for prokaryotes, and E for eukaryotes. And let's make the P red, right? And the E yellow. What we find is we find E, eukaryotes, and prokaryotes up here, no problem. Prokaryotes and eukaryotes all here. Evidence of them. Of course, when you're talking about single cells, a lot of times what you're talking about are their products that are left over, since they have no hard, bony material to leave behind. They can leave remnants, they can leave imprints, they can leave products, things that only they make, that we know today that they make here on the cell. Here's where we live up here. So here there's prokaryotes and eukaryotes living together today in life. And underneath for years and years and years and years, we see there's prokaryotes and eukaryotes living together, different. And the exact eukaryotes are different. As evolution went on, we can find that as we go further and further back, things get simpler and simpler. As you know, it's just a fact. These are facts. As we dig down deep into the earth, what we see, except for those places that have been turned over, as we discussed earlier, what we see is just these more and more evidence of prokaryotes and eukaryotes. Again, which prokaryotes, which eukaryotes, it changes as you go back into the fossil history. You know, you might find dinosaurs somewhere down here. You know, the T-Rex might be down here, right? maybe you'll find aquatic organisms down here more than anything else, and there are boundaries where things have changed, like major evolutionary changes. But what we do find is at some point you no longer have any evidence of eukaryotes. All you have are prokaryotic evidence. So all we have is prokaryotes, and different kinds of prokaryotes, to be sure. But the only thing left. Ooh, I used the wrong color. Sorry. So as we look down, as we go further and further down, all we see is prokaryotes. We have those stacks of rocks in Australia that you can go back and look at that had green, not green, we don't know the color, but probably green, cyanobacteria. So at some point there's cyanobacteria billions of years ago. And this is really not to scale, to be honest, because the layers would be much deeper than that, right? Because if this is 600 million years ago, 3.5 billion years ago would be, well, at least, 1, 2, 3, 4, 5, 6, 7 times deeper, but somewhere along the line down here you had cyanobacteria. And cyanobacteria is still around today, by the way. And that's a misconception in evolution, is that somehow, well, if cyanobacteria became chloroplasts and mitochondria, why are there still cyanobacteria around? Well, only one line of the species, only one group, one individual, a small population of the individuals need to move in a direction while the rest stayed the course for evolution to occur in one line, one lineage, and the other lineage continue on doing what it does. So don't get that confused, that just because something evolved from a common ancestor that necessarily means that common ancestor's dead or is gone. It probably is. I mean, in most cases it is. But it doesn't mean that it's here either. It's a matter of circumstances. Conditions change, and every different situation is going to be slightly different. Well, so this is the evidence we have so far. So it's 3.5 some odd billion years ago, cyanobacteria were around. There were no eukaryotes, no evidence of eukaryotes, no matter where we dig all around the world. Time goes on, more bacteria. That's all we find is evidence of bacteria all over the world. Then, and I'm talking about places like Australia, as different as Australia and Mongolia, and no matter where we look at these frozen deep places of the earth, we find cyanobacteria, but no eukaryotes, certainly no multicellular organisms. Then all of a sudden we start finding eukaryotes. Now these are eukaryotes 6 million years ago. But then that's not necessarily multicellular eukaryotes, right? These are eukaryotes that we know that as time went on, we got more and more multicellular organisms. And T-Rex wouldn't be down here, by the way. T-Rex would be up here somewhere because it's a eukaryote, obviously. It's a multicellular eukaryote at that. So it would be higher, not lower. All right, so anyways, cyanobacteria formed. These are all prokaryotes. Then eukaryotes showed up. We know that chloroplasts do photosynthesis. We know cyanobacteria do photosynthesis. So photosynthesis came before plants, before eukaryotes. We know that there are single-celled organisms, prokaryotes, that are doing cellular respiration down here as well. So that we know, those are things we know were happening based on the evidence that we have. We know that this was a hotbed of different organisms, right? Uh, so all over the world, in the oceans, in the water, lakes, in wherever there was water, there are these single-celled organisms all competing, right? We know they're competing. So we know they were probably eating each other. So we know that there was engulfing, there was one was eating another. How do we know that? Well, we assume that that's true based on what we see today. We see it happening today, and there's no reason to assume that it's not happening it wasn't happening then. If it's happening now, why wouldn't it be happening then? So there's these bigger cells would eat the smaller cells, as we see happening today. So there's a big cell, and here's a small one, a big and small prokaryote. Maybe this small one does cellular respiration really well. And this bigger one engulfs it. It does phagocytosis, the way we've studied it, right? But for some reason, that prokaryote, and let me go ahead and make it red, because that's the color we said we we're going to make it. That prokaryote can't be digested. Something went wrong. It has some kind of protection, or maybe the bigger prokaryote. Remember, these are both prokaryotes at this point. The bigger prokaryote just couldn't digest that smaller one for whatever reason. So now you have this prokaryote in here, does cellular respiration really well, surround a second membrane around it, and then this bigger prokaryote sitting there. This bigger prokaryote is really good at eating things. It can go around and eat things all day long. This little one is really good at digesting, at taking sugars and breaking it down, and oxygen, breaking down into energy, into ATP. So over time, this big cell is just going to keep going with that little, and it still has all its parts, right? It still has its DNA, it still has its ribosomes. It can still make copies of itself, but it's within a membrane. So over time, it's thought that this is the beginning of a new lineage. This is what will turn out to be a cell that has a mitochondria. This turns out to be the mitochondria that we have in our cells today, with all those different qualities. Now, I want to draw this out and just point out that there are other bacteria that are just like the one that was swallowed up. Maybe these guys never get swallowed up, and they're still around today. Or this is the ancestor of some of those aerobic, right? Those respiring bacteria that we see around today. These are the ancestors of those, where this becomes the progenitor, the first mitochondria that eventually turns into the mitochondria that we have today. Today, they're not bacteria. Today, it's not a single bacteria inside a cell, inside your cells. But they have bacteria-like properties that are inside our cells, even to this day. Now, if you can imagine that cell went on and found this little cyanobacteria, and then that cyanobacteria could do photosynthesis, and that eukaryote, and I should maybe use blue in this case just because, well, let me use yellow because that's the color we said we're going to use. And I don't know if this is a eukaryote already or if it's eukaryotic probably isn't in the eukaryote yet, comes along years later and starts to engulf a cyanobacteria. It already has a mitochondria, right? It already has a mitochondria in its cell, probably many. But as it moves on towards its ultimate eukaryotic destiny, it starts to surround the chloroplast, engulf it in what we learned is phagocytosis, remembering that the mitochondria is still there. Producing what eventually will be a chloroplast. So now it has two organelles. It has a mitochondria and it has a chloroplast. Well, this cell here takes off on its own. Before it engulfs, it takes off on its own. One of these many cells, because it's going to do cellular replication, right? It's over many millions of years, becomes the first animal cell. Where this one here, after many generations and millions of years, becomes the first plant cell. Notice that the plant cell has both mitochondria and chloroplast, where the animal cell only has the mitochondria. That explains what we see today as well. So this theory, this explanation, explains all the evidence that we have at our disposal, which is what a theory is. This is the evidence. This is what we see happening. This is all the evidence we have. And this theory of endosymbiosis explains, as best we can, the evidence that we have in front of us. So here you have let's go ahead and look at this description. This is the chloroplast. There's the circular DNA. Here's the internal membranes of the chloroplast that do photosynthesis. Here's the external membrane. It's very much like all our membranes. This internal membrane is very much like that of modern cyanobacteria, right? The structures found here, if you look at this structure here, look at a cyanobacteria, they're very similar. Here's mitochondrial DNA. It has mitochondrial DNA, also circular, right? The genes that are in here help it make copies of itself. Both of these make copies of themselves. This is all that evidence that we listed here in the Venn diagram. So if you could imagine that there's this pre-mitochondria bacteria, aerobic bacteria. It's a prokaryote that can do cellular respiration really well. Here's this cell. Comes along. It's big. It engulfs really easily, and it does. It starts to learn how to package its DNA in the eukaryote, right? So it has a nucleus. It engulfs the pre-mitochondria. This is theory now. We've never seen this happen. We don't have any fossil evidence of this. But what is this? Where are we getting this? We're getting this from the evidence that we have. So the cell engulfs the mitochondria. There's the mitochondria. There's the nucleus. Now from here, this cell would take off and turn into animal cells. So that's one sequence that led to animal cells, right? But one of its children, one of the first of, this being the ancestor of the cells, one of its progeny, one of its millions and billions of progeny that are going to be made as it does its cell cycle, it goes, so one of them goes on and eventually becomes animal cells that we see today. This, one of these engulfs then something that was a pre-chloroplast, very much like a cyanobacteria today. It engulfs it, and this becomes the first chloroplast. Again, they have their own DNA, they have their own ribosomes, they have a double membrane, the internal membrane being much like bacteria as well, the ribosomes being very much like ribosomal bacterial ribosomes are smaller. The sequences are very similar. The DNA and RNA sequences are very similar to bacteria that we find today. So this goes on and becomes a plant cell. This not only explains how you get all those similarities that we listed here, but also how we get plants that have both mitochondria and chloroplasts, but animals only have mitochondria. So this theory is beautiful because it explains a lot of the evidence, all the evidence that we have today. That's why it's accepted. So distinguish between two possible sequences of evolutionary change suggesting an endosymbiosis theory. Well, I just explained it all to you, so you should be able to write it in your own words. How does endosymbiosis theory account for the origins of the following mitochondria and chloroplasts? Again, you should be able to explain that. I just got done explaining it. What evidence of modern mitochondria and chloroplasts supports endosymbiosis theory? I just listed it for you. Comment how the fossil evidence of early life supports and contradicts endosymbiosis theory. Well, I just got done explaining that as well. Also, you can go back and reference the fossil evidence that are on the previous pages. If you find something, if you have your own thoughts, feel free to contradict anything I've said based on evidence, right? So anything you state, make sure it's based on something you've read here or something I've said, or is some piece of evidence something you've read somewhere else. So just make sure that you have evidence, right, based arguments. All right. Again, keep safe, stay away from crowds, and I'll see you soon.